I'm so excited because in this lecture we get to talk about the absolute most wonderful molecule on the face of the earth, DNA. That's right, deoxyribonucleic acid. That's what makes you, you, me, leaves, bugs, worms, bacteria. It's all about life. And if it wasn't for DNA, you wouldn't be sitting there learning about this. So in this lecture, we're going to learn about the structure of this beautiful molecule, how it replicates itself, and what happens when things go wrong, or it could be considered right depending on the situation, and that's called mutation. So we're going to watch some video clips. We're going to draw this molecule. It's going to be awesome. So here are three different interpretations of DNA molecules. And yes, it is a molecular microscopic molecule, but as you'll find out if you, for example, take the lab for this course, you can easily extract your DNA from your own cheek cells or from strawberries, onions, you name it, and actually be able to get enough of it there that it's gloopy and gummy and you can actually see it. So yes, it's microscopic, but yes, you can also see it if you get enough of it. And how much is there? Tons. You get about 3 meters of DNA packed into the nucleus of a single cell. So it's really just packed in there. It's awesome. So in reality, if you had an electron microscope, a DNA molecule might look something like this. And yes, it is called a double helix is the shape. It looks like a spiral staircase. And we know that Watson and Crick are credited with discovering the structure of DNA as being a double helix in 1953, won the Nobel Prize for it. But as you'll also learn, they weren't alone. A lady named Rosalind Franklin perhaps may have actually given them the idea that the molecule was a double helix structure in her work on crystallography. So if we took this DNA double helix here and we broke it on down to kind of look what it's made of, you'll see that it has kind of the two sides of the ladder. These are made of alternating phosphate molecules, that's PO4 you might remember, and a sugar, a 5-carbon sugar called deoxyribose, hence the name deoxyribonucleic acid. Now, if we unwound this ladder, you would see that the rungs of the ladder are made up of compounds called nitrogenous bases, sometimes just called bases. And these come in 4 types, and we're going to study these. So this is what DNA looks like in reality. This is what it's made out of. So we'll be going over each part of this. Now, you guys already know your DNA is found in the nucleus of the cell. Okay, so here's your nucleus, and that is where your DNA is packed into. So you might need to go back and review the different organelles of a cell if you don't remember what a nucleus is, but hopefully you do. So let's talk a little bit more about the structure here. Again, James Watson, Francis Crick. Those are the guys that are credited with discovering the double helical structure of DNA, but let's not leave out a very important woman, Rosalind Franklin. Again, it's packed in the nucleus of your cell, and this is the blueprint for life. This is what makes you who you are, because despite having really long strings of DNA, different segments of these bases, nitrogenous bases on the DNA, might code for a gene. And a gene codes for a particular protein, and that protein codes for a trait. So we're going to take an example of what happens. And in the subsequent lecture, you're going to learn exactly how you go from DNA to protein to trait. And there's another model of DNA spinning around, really nice. Again, it's a double helix, a twisted ladder made up of units. So again, the sides of the ladder are alternating phosphate molecules and deoxyribose sugars. The rungs of the ladder are made up of nitrogenous bases, and these nitrogenous bases we can call A, T, C, G. A stands for adenine, T for thymine, C for cytosine, and G for guanine. And we're going to take a better look at those right here. Okay, so in fact, you can take these A, T, C's, G's and break them into 2 categories. So we have purines. These are the larger of the 4 molecules. So these include the adenines, which we abbreviate with an A. And here is adenine right here. You know, it's a pretty little compound. Recall from chemistry that everywhere where there's a little corner in this ring structure, there's a carbon right there. And this represents double bond. And guanine is the other large one. So here's a guanine. And this is pretty cool because look how similar adenine and guanine are to each other. In fact, not a whole lot of difference there. You have a double bond to an oxygen here, whereas you have an NH2 right here. Other than that, very, very similar compounds. Then we have the pyrimidines, and actually this should be, okay, and then we have the pyrimidines, and these are the smaller of the four, and they include thymine, which we abbreviate with a T, and cytosine, which we abbreviate with a C. So here is thymine, here is cytosine, and again, look how similar these compounds are to each other. Not a whole lot of difference here. There's a double bond to an O there, and instead here there's an NH2, and this one's missing a methyl group, but other than that, the exact same compounds. So you can kind of think, well, maybe during the evolution of life on Earth, we had chemical evolution where you get slight changes in some of these nucleotides, and boom, you end up with what you need to get life started. So nitrogenous bases, very cool. Again, we have purines, we have pyrimidines. The purines are the A's and the G's, adenines and guanines, and the pyrimidines are the smaller groups, the thymines, the T's, and the cytosines, the C's. Now, here's what's even cooler. We have what's called a base pairing rule, and that is, this is a law of life here. C always likes to pair with G, and they form 3 hydrogen bonds between those two compounds, and A always likes to pair with T, so they form 2 hydrogen bonds. So anytime you have a G on one side of the ladder, you'll get a C on the other side, and vice versa. And anytime you have an A on one side, you'll get a T on the other, and vice versa. And they're held together with hydrogen bonds. And that's really important that we're held together with hydrogen bonds, because you might recall that hydrogen bonds are weak bonds. They're easily broken and easily made. And that's going to be critical for when we replicate this DNA using what's called semiconservative replication, which we'll go over in a minute. But if it weren't for the fact that we can easily break and form those hydrogen bonds, DNA could have never split, which means it could never have replicated, which means you wouldn't be sitting here listening to this lecture. So many things had to happen for life to occur, yet we have this phenomenal diversity of life. Life's cool, isn't it? I like it. I like it. I happen to be living. Anyway, so C with G, A with T, that's called the base pair bonding rule. And it's fundamental to whether you're a human, or you are a bacterial cell, or you are a plant, or you are a fish, or whatever it is. We all use that same genetic code. DNA is the same, and in fact, it's switchable. I can take a gene from a human and stick it in a bacterium, or vice versa, or a plant gene and stick it in a human and have it work. I mean, it's amazing. It's one universal code for all life on Earth. Okay, anyway, just had to get that out. Okay, now the other thing about DNA, the two sides of the ladder are what we call anti-parallel. That means they go in opposite directions. So if you look at this picture, you'll see here that one end is what we call the 5' end. And if you follow that all the way down, the other end of that side is the 3' end. And the opposite is true of the other side of the ladder. So if we started with 5' on this side we're going to start with a 3' end and follow it down. It will become a 5' end at the other end. Now, what do we mean about 3', 5' doesn't seem to make any sense? Well, remember those deoxyribose sugars. Remember OSE, anything ending with OSE is a sugar. Well, there's 5 carbons, so we can number them 1, 2, 3, 4, 5. And depending on which of those 5 carbons is facing up or facing down, it'll be either the 5th one, which will make it a 5' end, or the 3rd one, which will make it a 3' end. And so that's where those terms come from. They're referring to which carbon is up or down on the thing. But what you need to remember is that one goes from 5' to 3', the other side of the double helix goes 3' to 5'. So they are anti-parallel. They run in opposite directions. May not sound important now, but when we learn about some of the details of DNA semi-conservative replication, you'll find that it is, in fact, important. Okay, so this allows for DNA and RNA to form long chains of nucleotides. And again, you can see in this diagram, notice C's and G's are together and A's and T's are together. Okay, so before we go on to learn how DNA replicates, let's watch a little video clip. So I'm going to pause this for a sec and get up this video clip, and that'll go over a 3-dimensional view of what DNA looks like. And thanks to computer animation, which they didn't have when I was going to school, it's pretty cool. We'll give you a nice visual.
DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
Was that cool or what? So that gave you a nice 3D visual of the structure of DNA. Now what we're going to learn about is what's called DNA semi-conservative replication. So this is the way DNA replicates itself. So think back to that cell cycle, the life of a cell, and you recall that the M phase is where mitosis occurs. That's where cellular reproduction occurs, where a cell will split from 1 into 2 in the case of animal cells, or build a cell plate in the middle to create 2 cells in the case of plants. Well, that was only one part of the life of a cell. You remember gap 1, gap 2, those were the interphase kinds of metabolic activity phases. And then there was an S phase. And it's during that S phase that we undergo DNA semi-conservative replication. That's where the DNA itself is going to replicate in anticipation of the cellular reproduction coming down the road. Now, when they were first trying to figure out how DNA replicated, there were a couple of different ideas. They thought, well, maybe you have a parent and then suddenly out of nowhere you get 2 new ones, but that's not the way it happens. It happens like this, where you have the parent DNA, those hydrogen bonds are broken between the bases, the nitrogenous bases, so you end up with one parent strand over here and one parent strand over here. Fair enough. Well, then there's going to be free nucleotides, free nitrogenous bases floating all around the nucleus. So the A's, the T's, the C's, the G's, they're just constantly floating around your nucleus by themselves. And when your DNA splits, now those babies are able to come and, using the base pairing rule, form the new strands of the DNA molecule. So you'll see here, here's your old strand. So if you had a G on the old strand, a free nucleotide cytosine will come along and base pair with that. Boom. And there's another C, so here's a G from that was floating around the nucleus, boom. If you had an A here, a T here, boom, 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 and you get this new daughter strand forming along the parent strand. And on the other side, same kind of thing. Here was the parent strand. If you had a C here, a free G will come there. If you had a free G there, a C will come there, etc. And so you end up with 2 new daughter DNA molecules being formed from that one parent strand. So it's called semi-conservative because in each new daughter molecule, one strand is from the original parent, so it was conserved, and one is new. Now, there's a little more detail into actually how this happens. One new strand will be what's called the leading strand, and will go from one end to the other. The other is going to be called the lagging strand, and will go from the opposite direction up, and then you'll get some things needing to happen to fill in the gaps and things like that. But you'll end up with 2 brand new DNA molecules, and they'll be identical to the parent DNA molecule. So we have conserved our genetic blueprint, unless there's a mutation, which can always happen. So let's watch a video explaining how DNA semi-conservative replication works. So I'm going to pause it for a sec, be right back.
In order to make a new copy of a DNA molecule, a process called semi-conservative replication takes place. The double helix is firstly untwisted by this enzyme here, called DNA helicase, and it does this by breaking the hydrogen bonds between the bases. These free nucleotides then pair up with their complementary bases over here through hydrogen bonds. Now other enzymes, called DNA polymerase, link these new nucleotides together by covalent bonds. So now each of the new DNA molecules is made up of one old strand and one new one. And that's why we call this semi-conservative replication.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So how cool is that? It goes as fast as a jet engine, yet this is going on in each of the cells of your body all the time. It's amazing. This is DNA replication. So again, let's just sum up real quick. You get a parent strand of DNA, enzyme, because that's what enzymes do. They like to digest stuff. It's going to come along, break those hydrogen bonds, that's DNA helicase, and it's going to split this molecule into two. So you get one parent strand over here, one over here. Free nucleotides are going to base pair on one side and base pair on the other in opposite directions, and boom, you end up with winding up and two new DNA molecules. I simplify, there's a lot of enzymes involved, but you get the gist for a non-majors class. Cool or what? Okay, so now let's talk about a little bit about when things go wrong. So normally C with G, A with T, but sometimes something in the assembly line doesn't work, and you get what's called a mutation. And there are many different kinds of mutations. Oftentimes a mutation in your DNA or the chromosome the DNA is in can be a really bad thing if it happens in the sex cells that are going to give rise to a baby, or happens in, say, the fertilized egg that will become the baby. It often produces a genetic abnormality. It can be very bad. A lot of times, in fact, I'd say most of the time, these mutations are neutral, and you'll learn when we talk about protein synthesis why that is. But occasionally, having a mutation can be a good thing. So in the next unit, we're going to be learning about evolutionary biology. And the only way life on Earth has been able to change, i.e. evolve, is because of mutations happening in the genetic code. If that mutation happens to produce a protein that produces a trait that makes that organism better adapted to a changed environment, that individual might be more likely to survive, reproduce, and pass that mutation on to their offspring. Over time, you can have an entire population that is better adapted to its environment. So mutations can be good, they can be bad, and they can be neutral. So here's an example of a type of mutation. Here's your DNA, and let's say instead of getting an A here, I got a C. Well, that would be a big oops, and that would probably cause some major problems because now our base pairing rule is not doing what it's supposed to do. Okay, so mutations are errors in the copying of DNA, but this is the primary source of genetic variation that natural selection operates on, and overall mutations are rare events. They have actually gone and calculated a mutation rate 10 to the negative fifth to 10 to the negative sixth loci per gamete per generation in eukaryotic organisms like ourselves. So what that means is if you take any gene at any locus, any location on a chromosome, inside a sex cell, each generation there will be a 0.00001 to 0.0000001 chance that you'll have a mutation in that area. Now, don't worry about memorizing that. What that means is mutations are rare. But over an evolutionary time scale, it's pretty certain that any one gene will eventually experience a mutation event. So rare, but they happen, and it's enough to drive evolutionary change, which is so cool. Okay, so again, most mutations are probably deleterious. That means they have a negative effect on the organism, often resulting in a miscarriage. So many of you might not have known that most miscarriages out there are due to mutation events that have made that embryo probably prone to a genetic abnormality. And in most cases, those are lethal, and the embryo gets miscarried instead of being born. But again, they can also be neutral or beneficial. Now, usually things don't end up with mutations, and the reason why is in a healthy organism with an intact immune system, we have special enzymes that are the proofreaders of this DNA semi-conservative replication. In other words, as that DNA is replicated, proofreading enzymes will come along and see if there's an oops. And if there's a mutation, they will take out the mutation and repair it with what's supposed to be there. How cool is that? Okay, so in other words, it's a very rare event that that mutation gets left, that it escaped the proofreading mechanism. It's kind of like, you know, you have an English paper to write and you proofread it, you catch all the errors. Once in a while you might have missed something and that error gets turned in and you get a bad grade on it or whatever. So one of these proofreading enzymes is called DNA polymerase, and it goes through and it proofreads each of those nucleotides, each of those nitrogenous bases, H, C, G. It's going to go through and make sure that it was correct, and if it's not correct, it's going to repair it. And so this diagram here, you can kind of see, here's your DNA polymerase coming along during the semi-conservative replication, and if it finds an oops, so it's going to, imagine this little thing is the oops here, it's going to take out that bad nitrogenous base, and in the case of if it's a big mistake, it might take out a bunch of bases and repair it with the correct one. So, and then it keeps moving along its proofreading merry way. Okay, so the actual, by the time the proofreader has gone through there, by the time that DNA polymerase has gone through, your final error rate is 1 per billion nucleotides. So, in other words, mutations are rare. We have pretty good mechanisms. Now, you might have noticed that as people age, they're more prone to problems like cancer. Well, guess what? As you age, your immune system breaks down and you're not as likely to produce the correct proofreading enzymes to do the proofreading. And so it's more likely that some of these mutation errors will get missed and can result in things like cancer. So keeping your immune system up is a good thing. Exercise helps with that. So lots of different kinds of mutations. One common kind is what's called a point mutation or a base mutation. And there's two categories for these. We have addition-deletion mutations. And this is where a single nucleotide base either got deleted where it was supposed to be there, or it got added in when it wasn't supposed to be there. So I'll give you some examples of these on the next slides. But so for example, let's say your DNA sequence you were supposed to get was Gattaca, G-A-T-T-A-C-A. Well, maybe instead of getting that first A, it got deleted. So now you're G-T-T-A-C-A. Or maybe it got duplicated. So you're G-A-A-T-T-A-C-A. That's called a point mutation or a base mutation. A single nucleotide gets added or deleted. Now, sometimes it can be several in a row that get added or deleted. And sometimes you get what's called a substitution base mutation. And in this case, you don't get something taken out or added in, but you get the wrong base put in there. So back to Gattaca, G-A-T-T-A-C-A, maybe instead of that first A, I get a T. So now it's G-T-T-T-A-C-A. That's called a substitution base mutation. Okay, now sometimes these result in what are called silent mutations, and you'll learn about this when we talk about protein synthesis in the next lecture. In this case, you did get a substitution, but you end up still with the same amino acid being produced that you were supposed to get, which gives you the same protein, which gives you the same trait. It's a silent mutation. You got a mutation, but you still get what you were supposed to get in the end. And in next lecture, you'll learn how that happens and why that happens. So I don't want to get too far ahead of myself, but just know that many mutations produce neutral change because you still get what you were supposed to get, even though the DNA changed. This is a cool one, transposition mutations. These are where they're sometimes called jumping genes. This is where a section of DNA or even a single base of the DNA literally jumps onto another chromosome in the genome. How cool is that? I mean, oftentimes it's not very cool because you end up with a lethal disease, say Philadelphia syndrome, where a section of chromosome 9 and chromosome 13 swap. You know, not a good thing. It's a lethal disorder. But it's interesting to see how this happens, that you can actually get big chunks of DNA, long sections of DNA, move from one part of the genome to the other. And then you can actually have mutations where the whole chromosome mutates. You get maybe a piece of the chromosome breaking off and inserting somewhere else, or the chromosome duplicates itself, or all sorts of things can happen. So here's an example of some chromosomal mutations. So this is called a deletion chromosome mutation, not to be confused with deletion point mutations. This is where, like, let's say this is the chromosome you were supposed to get, and here it represents a huge chunk of DNA with lots of genes in it. When you go to replicate your DNA and your chromosomes, boom, a whole section of it got taken off. Well, you just now are missing a whole lot of genes that you were supposed to get. Not very good thing in the end in most cases. Here's an inversion chromosome mutation. This is where literally the section of DNA that should have been up here breaks off and inserts somewhere else, like at the bottom of the chromosome. Yikes. Not good, because now your genes are all out of order. Here's a duplication event where this whole section of DNA got duplicated and stuck onto the end of the chromosome. Now you get twice as much of those genes as you were supposed to get, which means you get twice the amount of protein you were supposed to get. Oftentimes not a good thing. And then here's that transposition, or translocation as it's sometimes called, where literally that DNA section, boom, jumps and ends up on chromosomes that it's not supposed to end up on. So all sorts of weird things can happen. So here's some examples. So let's talk about those substitution mutations. Let's say, for example, here's your DNA sequence, GCA, AGA, CGG, TGC. Let's say that's normal. That's what you were supposed to get. Well, in the next lecture, you'll learn that in order to create proteins from genes, in order for your DNA to code for what protein you're supposed to get and what trait you're supposed to get, you have to make an intermediate molecule called RNA. And as you'll learn in more detail, RNA is very much like DNA, except that instead of thymine, the T, it uses a U, uracil. And so you get RNA from the base pairing with DNA. So if I had a G in the DNA, I'll get a C for the RNA. If I had C in the DNA, I'll get a G in the RNA. A will give me not T, but U, etc., and all the way down. So here's my RNA. Well, every 3 bases of RNA codes for a particular amino acid, and you'll learn how that happens in the next lecture. So CGU, if I looked it up, I'd find that, you know, when the DNA sequence was GCA and my RNA is CGU, I'll end up with the amino acid ARG, which is arginine. And then the next 3, this is supposed to code for an amino acid serine, this is supposed to code for alanine, and this is supposed to code for threonine, etc., all the way down. And basically I'm trying to form acetic acid here. Well, anyway, let's say this is what I was supposed to get. Don't get too hung up in what all these mean. You'll learn about that later. But if a substitution mutation happened in the DNA, then instead of getting an A here for adenine, I got a G here for guanine. Well, now when I go to make my RNA, I can't get the U that I was supposed to get because now it has to base pair with a G. So I get a C. Well, I just changed what amino acid. Instead of serine, now this codes for proline. So I just got a different amino acid. And in many cases, that could be a problem because that's going to change when I go to take these amino acids and fold them up into the protein they're supposed to be. That can actually change the protein or make it not function at all. So in many cases, that's going to be a bad for the organism. Sometimes it won't change it enough that it causes too much of a problem with the protein, but many times it does. But that's an example of a substitution mutation where instead of getting this DNA base I was supposed to get, I get a different one, which causes a different amino acid to be produced. Now that's bad enough, but what really tends to cause problems is when you get what's called a frameshift mutation. And a frameshift mutation happens when you get a deletion or an addition, or you can call it an insertion of a base where that wasn't supposed to happen. So here's the same sequence that we just went over. Let's take a look right here. So here it was supposed to go GAC. Well, if I get an insertion of a T between that A, that G, and that A, that throws everything off. So if I'm taking a T and sticking it right there, Well, now I have GT, and then that throws that triplet, so that now that A is part of this next triplet. Remember, every 3 codes for an amino acid. So AGA should have given me RNA that said UCU, which coded for serine. Now instead, I have AGT causing a UCA. Serine, okay, still same amino acid, not so bad, but now everything's shifted. It's shifted over a space. So now the next triplet, instead of CGG coding for GCC, coding for alanine, I get ACG coding for UGC, which codes for cysteine, which throws the next 3 off, which codes for histidine instead of threonine. So now everything downstream is off. I get completely the wrong sequence of amino acids, which means I get completely the wrong protein, which means I don't get the trait I'm supposed to get. This usually causes genetic abnormalities, generally not a good thing. And here's some examples of chromosomal mutations. So remember that a chromosome is a bunch of DNA packed into these chromosomes, right? You can have 1 chromatid or duplicated, 2 chromatids is 1 chromosome. You remember all that. Well, let's say this is my chromatid, and every section here represents multiple genes, many genes along that chromosome. We'll call them those sections A, B, C, D, E, F. But remember, these are many different genes within each of those sections. Here's some actual photographs of human chromosomes here. So imagine we took one of these chromatids and it ended up breaking. This happens. Things go wrong with the assembly line when you're replicating your DNA and replicating your chromosomes. So in this case, this would probably happen around mitosis, where a whole section of that DNA that's in the chromosome broke, and so you get a section here separated from this section, separated from the rest of it. Well, when you go back to delete that section B, and now these get stuck together, you have the wrong order of genes. Very bad. Or maybe instead this thing broke right here and right here. Now you have this section. Bad enough, but now when it goes to repair itself, you can actually have a section completely inverting. So this is an inversion event. So instead of getting A, B, C, D, E, F, I get A, B, E, D, C, F. Anyway, stuff's bad. Now my genes are out of order. They're not going to be created right. The protein order is not going to be right. Things are bad. Or you can actually get whole sections duplicating. So instead of A, B, C, D, E, F, I get A, B, B, C, D, E, F. Well, that, again, is going to produce what's called upregulation, where now I have so many extra copies of the genes that I'm producing so much more protein, and that could be really, really bad for my organism that I'm creating, whether it be a human or a plant or whatever. Okay, so that's it. I hope this makes sense, and if not, it's probably going to make sense when we put it all together in the form of learning about protein synthesis. Till next time.